Okay, so I can now copy the link. Meena, are you there?
Yes.
Oh, because your video is frozen. Oh, okay, good.
Yeah.
I was concerned for a second.
No.
Okay, so I will just send the YouTube link to everybody. Okay, and I guess I can start letting people in. Right. Okay, welcome everybody to our last day of the workshop. We are very glad that you are joining us today, and we have a very exciting program. So before we start, I will let Ryan say a couple of words about what can be done after the workshop if you would like to continue working with the material of the workshop on your own. So Ryan, go ahead.
Great. Thank you, Olga. I'll share my screen here. Great. So as everybody knows, we've been using RStudio Cloud to work on the projects. Maybe some of you have been working on your own machines, but I think most people have been using RStudio Cloud. And I put some additional instructions on our GitHub site here to help your next steps on what you can do after the workshop. So at our GitHub site, you've probably been here quite a bit. If you scroll down to the bottom here, I have some post-course notes here. And I have some notes here about continuing to use RStudio Cloud after the workshop. Just to be clear, the actual workspace we've been using for the project, that's going to be taken down to sort of prevent the continued use of the computer hours, which were being billed for. But you can continue to use RStudio Cloud on your own free account after the workshop is over. And I want to show you how you can make sure you retain all of your projects in your files. I have instructions here on the GitHub site, but I'd like to show you live how that works. So if you go to the workshop page for RStudio Cloud here, you'll see something that looks like this. You'll see your different projects that we've been working on. It might be a little confusing that you can see that these come in pairs here. These link to the same place. The one that has the continue button is sort of just a weird little link to the original project. That will take you to the same place as your project that you've been working on. So think of these pairs as really just one entity. And if you look to the right of these projects, you'll see this ability to move the projects. And what we want to do, for example, is take the projects and move them to your workspace. Okay, so let me show you again. Go to one of your projects, look for this little move icon here, click your workspace and click OK. This is going to be after the course, by the way. So go ahead and keep working here for today. But tomorrow, for example, when you move your project, you'll just click on that this move icon, and then you can move it to your workspace. And then if you go over here to the little hamburger menu up here, you can go to your workspace and you'll see these projects live here in your workspace, and you can click on them and continue to work on them. One note is that you probably have the free RStudio account. You get 15 hours a month free to use those projects. So you are limited in terms of the compute hours you can use to access your projects. But nonetheless, this is a way you can continue working on them. I also wanted to note here that you can install R on your own computer as well. Maybe some of you have done this. If you haven't, highly suggest that you do that because you probably have a lot more memory on your computer. And obviously you can use it as much as you like. I've prepared some slides here to give you some pointers about installing R and RStudio on your own computer. So go ahead and click on that. And there's a slide deck here that gives you some tips on doing that. Okay. One last note on the RStudio Cloud instances, particularly for today's workshop, we're using computers with more RAM because that's what's required for these files. So these day 4 projects require more RAM that we've configured on this particular workspace for the workshop. On your free account, you don't have as much memory. So running these examples in RStudio Cloud probably will not work or will not work well on the free account. But the other projects, day 1 through 3, should probably work just fine. So just want to give that pointer and suggest moving to installing R on your own computer to avoid those problems. So if you have any questions, go ahead and post them. I'm going to put my email address in the chat here. Feel free to reach out to me if you have any issues installing R or working with RStudio Cloud after the workshop. One last pointer, I'm going to shut down the workspace on Monday night, so please move your projects before Monday so you have access to them. So that's it. Thank you very much.
Well, thank you very much. So I do hope that you will be able to continue working with all the materials of the last 2 weeks. So now let me go ahead and share my screen. So we'll move on to the presentation for today. And so today we will have 2 parts. So the first hour and a half, we will talk about software MSSTATS, which implements statistical methods for relative quantification of in mass spectrometry-based proteomics. And the second part, we'll talk about Cardinal. And this is a software for mass spectrometry imaging. And so there, the interesting thing is beyond just working with imaging, also how to work potentially with large files and raw files. So there was a question in the beginning last week about how to deal with raw files. So there will be some discussion of that. And so in the first day last week, we talked about some basic R and how to create basic data structures and how to manipulate them. On the second day, we talked about an environment which was developed on top of basic R, so which is informally called tidyverse, and so using ggplot as one library to do data visualization. So this is the library which was created on top of base R. So similarly, yesterday, Meena was showing how to use pretty basic functionalities in R to do some really simple statistics. And so today we're going to talk about things which are more advanced. So this will be already analysis of real mass spectrometry data. And so therefore we need a little bit more memory, right? And we need a little bit more functionality. So I just want to once again encourage you, if you feel that you have been overwhelmed by the last 3 days and you have very hard time keeping up, skip this part, go straight to your TAs who are available right now, work with them through the exercises to make sure that the beginning part of the first days you really have it down, because I think it will be best use of your time. And of course, the presentation will be recorded. As a matter of fact, let me start recording right now. Here we go. So now we should be able to, and of course it's broadcast live as well. Okay, so just as yesterday, what we will do for the part MSSTATS is 2 parts. So very briefly, I will give a crash course on MSSTATS and introduce the concepts behind that. And then Meena will be doing the examples, live demo, and then there will be a portion for you to try it out if you're interested. And so let me start with that. And the very first thing I would like to do is to acknowledge the developers of MSSTATS. So MSSTATS is over 10 years old. We have quite a few contributors, and Meena was the lead developer of MSSTATS throughout this project, as well as Ting, Mateusz, and Sung Heng, who made really substantial contributions. And we have quite important contributions from other contributors as well. So we are very grateful for their work. So let me talk first about what MSSTATS is. So I have the Zoom window, which blocks part of the screen. Let's see if I can eliminate this somehow. So MSSTATS is a package which is written on top of base R. So this is essentially software which is written on top of base R in a similar way as, for example, ggplot and other libraries that we've been using in the last 3 days. So one of the main questions that on which MSSTATS focuses is essentially which proteins change in abundance between conditions, which proteins are regulated. And in fact, MSSTATS is not just a package, it is by now an ecosystem which has many functionalities beyond just differential abundance. But today we will focus on that. But let me tell you just briefly what are the overall functionalities, just so you know. So when we want to make inference about which proteins change in abundance, MSSTATS will consider complex designs, for example, multiple factors and treatments, paired designs, time course designs, and combination of them. We can also estimate the abundance of each subject on a relative scale that is comparable between runs. So essentially have one estimate of protein abundance per biological replicate and time point or per mass spectrometry run. And this can be useful, for example, if you want to plot a heat map of these abundances or do some kind of classification method with that. So MSSTATS works with label-free and label-based experiments. So the base MSSTATS works with experiments which consider chromatography-based quantification. So data-dependent acquisition, data-independent acquisition, SWAST, PRM, targeted selected reaction monitoring. A additional package, which is a part of the ecosystem, it works with TMT labeling. We unfortunately don't have time to talk about this today, but there will be other opportunities, I'm sure. We also have a beta version of implementation, which looks for differential post-translational modifications. A very important aspect of MSSTATS is that we try to make it compatible with as many tools as possible which work on processing raw data. So there was a question last week about how do we deal with raw data in R. Sorry. So for the purpose for mass spectrometry and proteomics, we actually don't deal with that. What we do is we work with tools which have put a lot of, where developers put a lot of effort to read raw data, find peaks, identify peaks, quantify peaks, map peaks between the runs, and so on. So there are plenty of tools which are both open source, free, and commercial. And we try to work with as many of them as possible. So we work with Skyline, MaxQuant, OpenMS as far as open tools, right? And then Spectronaut, Proteome Discover, and others. And some of these tools, such as Skyline and OpenMS and Spectronaut, export the data directly into a format which is compatible with MSSTATS. But what we also offer is our own converters, where you just take standard output from these tools, and the converters which are part of the MSSTATS ecosystem will not just format the data into a form which is compatible with MSSTATS, but also do additional cleaning, for example, removing decoy peptide identifications if you had target decoy in DDA, and so on. This said, you don't have to use our converters. You can just have your own data processed in your own way and formatted as a table, and MSSTATS can work with that as well. And of course, the core of the software is the statistical analysis. So yesterday we talked about t-test and p-values. So since we are dealing with more complex designs, we need more complex models than just the t-test. And so MSSTATS considers that. And part of the work is things such as normalization, treatment of missing values, treatment of outliers. And a lot of work is invested into making sure that numerically our estimates are actually stable. And in addition, we have other things such as functionalities for system suitability, quality control, limits of detection, limits of quantification, and determination of sample size. So there is a lot that can be said about that. I have very little time, so I will just try to do the minimal work. So this is how a statistician like myself would view a quantitative experiment. So this is where data acquisition happens. And I would like to argue that a lot of statistics happens before collecting the data and after collecting the data. So usually we tend to focus on this part, but I would like to argue that this part is just as important. And what I just described in terms of just what can be done. So here, this is a list of different packages. All of them are packages, all of them open source, which are part of the MSSTATS ecosystem. And in addition to the packages, we also have a resource for storing the data, and I will mention this briefly in the end. So let me very quickly give the overview, then I will focus a little bit on statistical modeling, and I will talk about community resources. So first of all, what happens before we collect the data? Well, many things, but today I will just focus on experimental design. So we need to understand what are the goals of the study, what are the biological replicates, what are the technical replicates. And so here, for example, we can handle designs which have different cell lines with different oxygen treatment at different time points and cultures. MSSTATS can handle that. Here's another example where we have controls quantified at one time point, and then this is cancer patients quantified at multiple time points. So MSSTATS can handle designs like that. So a very important part at this stage is the annotation, because to make sure that our statistical models reflect the experimental designs appropriately, well, we need to communicate to the software which run corresponds to which biological replicate, which technical replicate, and so on. So this is a really important part here. So the question is if this works only on proteomics data, and what about metabolomics data? In principle, there is no reason why it wouldn't. In practice, we haven't had a chance to actually benchmark this and test. So if you are interested in applying this to metabolomic data, we will be happy to work with you on that. We just haven't had an opportunity to do that. So for the moment, I'm really talking about proteomics experiments, mass spectrometry-based proteomic experiments. Okay, so after the data are collected, as I mentioned, the very important first step is data processing, which in our case happens through converters in part, such as removing decoys, maybe using if your identifications of MSSTATS spectra in DDA have some cutoffs, like a Q value cutoff and so on, there can be filtering based on that. And removing, for example, redundant peptides. Sometimes in the same run, the same peptide ion has multiple peaks. So cleaning that up and so on. But then the second part of data processing, which is just as important, and this is what happens already in MSSTATS, is data visualization, QC, and normalization. So this is, just to be clear, an example of the format of input format that MSSTATS takes. So this is a long format, like what Meena described. So each row here corresponds to a peak in the LCMS space, right? So here, for example, for this protein, this peptide with this precursor charge, so this is an SRM experiment. So this fragment, this product charge, this experiment has heavy labeled reference standards, so we can have that. Comes from condition 1, biological replicate A, run 1, and this is the intensity of the peak. So essentially each row corresponds to the intensity of the peak, and all of these columns are the annotations that correspond to this intensity. And so what I was saying earlier, that it is really important to reflect the experimental design. It happens in the condition and bioreplicate space. We need to make sure that we correctly explain which run corresponds to which condition and which biological replicate. Okay, then another very important step, and this is also came up yesterday already, is normalization, because it is currently common that, especially if the experiment has many replicates and runs over many runs, that there are differences in the overall intensity of the signal. And this can be due just to some drifts. It can be due to different operators. It can be due to the batches of biological material. And so MSSTATS will make plots like that. So here, each row, the x-axis corresponds to a mass spectrometry run. The y-axis summarizes all the log intensities in this run across all proteins, across all features, and so on. And vertical lines separate conditions. So this experiment has 10 time points. And so we have 3 replicates within each time point. And so you see here that, yeah, so it kind of happens to be low in earlier time points and somehow higher in the later time points. And the question is it biology or is it technology? And the truth is that it can be both, right? So we don't know. So if we have no other information about the experiment, what MSSTATS would do, it would equalize the medians of these box plots between the runs. But this is actually a non-trivial thing to do, because this assumes that the majority of our proteins do not change in abundance between conditions. So the medians are roughly stable, and whatever individual proteins change, there's only a small number of them, and they do not affect the overall signal. So this is true, for example, in DIA experiments or in shotgun DDA experiments, which have many proteins, because then we expect that many proteins will not be regulated. Well, turns out this experiment actually a targeted SRM experiment, which has only few proteins, and they were handpicked so that they're likely to be changing. So this normalization will be a really bad idea. And so in this case, well, because of that, this experiment also included reference standards. And so if the experiment has reference standards, MSSTATS will make a plot like that, where the left panel corresponds to the references and the right panel corresponds to the endogenous peaks that we're interested in. And so now we see that the references, they have some variation, but not as much as here, right? Not as much as endogenous. And so MSSTATS would equalize either the medians or the entire boxes of the reference peptides and apply these shifts to the endogenous peptides. And so now we see that here, we did not remove the biological variation by normalization, right? We corrected it somewhat, but we still preserved the signal. And in my experience, this is one of the most crucial steps in the entire analysis pipeline, because assuming that there is no overall no change in protein abundance, when in fact there is one, either because you have very few proteins or because the experiment was such that most proteins are regulated for some reason. So this type of assumptions need to be very carefully considered. As far as MSSTATS, we propose different options. So the median normalization and also the normalization based on references. So you can have reference counterpart for each endogenous peak, as is the case in SRM. We can also have just a few global reference standards, which can be equalized. So after this is done, well, the very important part is statistical analysis, which I will focus on next. But let me just give you the upshot here. So the upshot would be that for each pair of conditions or each pairs of time points or anything you're interested in, we will be able to calculate the log fold change, as we discussed yesterday, and the p-value adjusted for multiple testing. And so MSSTATS at the end of the day can produce volcano plots if you are interested in only one comparison. If you are interested in multiple comparisons, for example, like here, where we have 10 data points and we may want to compare time 2 to time 1, time 3 to time 1, and so on. So then we can also display these results as a heat map where here the rows are proteins, the columns are pairwise comparisons, and red means significantly up, blue significantly down, and black no change. So it kind of allows you to see how proteins evolve across time as a group. And the last thing, experimental design, because now that we finished the analysis, we can use this information to inform subsequent experiments. And so in particular, we can quantify how much biological and technological variation we had in our current data. And this can allow us to make projections for the next experiment if it looks like our current one, then, and if we're interested in a particular value of fold change, what would be the minimal number of replicates per group. So this is part of the functionalities in MSSTATS. So now let me focus on statistical modeling just very briefly. So what actually is happening with the statistical models? And so the really big aspect of what we do is the fact that we want to work with different data processing tools. And different data processing tools make very different decisions as far as what is the abundance of a peak or the intensity of the peak, what is a missing value, and so on. And so here's one example. So this is a data set from Jürgen Cox from the LFQ paper a while ago. And this data set was processed with Skyline, MaxQuant, and Progenesis. And the histogram here summarizes the log intensity of all the features in this data set across all proteins, across all runs, everything. And so you see already that the location of these histograms is different between tools. Progenesis tends to report smaller values of intensities as compared to MaxQuant and Skyline, which is not a problem because we're interested in relative quantification and not in absolute quantification. But also you can see that, for example, Skyline has this bar. So this corresponds to 1,300 zeros in the data. But MaxQuant doesn't have this bar, so MaxQuant doesn't have zeros. On the other hand, Skyline only has 33 missing values, but MaxQuant has 38,000 missing values. So you see the different data processing tools. Well, when the peak is nicely resolved, I think most data processing tools will do similarly well. But when the peaks become, low intensity, overlapped, or have some interferences, then the differences between tools become more apparent. And so MSSTATS tries to kind of read between the lines and understand the meaning of those zeros and A's and small values and how to handle them. And in particular, we have a cutoff, which is empirical cutoff that we select, where we say below this cutoff, we don't trust the values. And so they can be zeros, they can be some small values here, they can be NA in the case of max quant, and we will view them as missing for reasons of low abundance. So in statistical language, it is called censoring. There are some exceptions. For example, Skyline only uses NA when you have two peaks which are present, but they're overlapped a lot. And so then it's not reasonable to assume that they're low abundant. So we just leave them as NA. Also, if you have missing values in reference peptides, there is no reason to assume they're low abundant. So we also leave them as NA. But otherwise, we view them as censored. And so this is a data structure that MSSTATS uses. So here the columns are runs, which are grouped by conditions, by subjects. Maybe you have multiple technical replicates for the subject, and we have different features. And so some of these values we now view as censored because they are below the cutoff. And so the challenges for us are to represent experimental design. So the pink part correctly, represent the variation in peak intensities, and estimate the variance components. What is the difference between assigning a value 0 or as a missing value? Hold on, I will answer this in the next slide or in a couple of slides. So actually right now, here we go. So let's talk about that. So what's happening with those censored values, right? So we just said that we kind of postulate that the values below this vertical cutoff, they are not reliable for reasons of low abundance. So what does it mean for us? Well, frequently these proteins we want to, so by the way, again, just to remind you that our goal is to determine which proteins change in abundance, meaning we want to make conclusions at the protein level, right? And not at the individual feature level. So we will be considering all the features which correspond to this protein, right? So this can be different peptide sequences or different ions of the same peptide, or different fragments if we work with DIA, right? Or different transitions if we work with SRM. And so if we look at all of the values which correspond to one protein, okay, so this is just a cartoon. So we have two mass spectrometry runs, 1 and 2. So let's say those are healthy and those are disease. So let's say that this is our limit of quantification. This is our threshold. So this value is not visible, right? So if we do nothing, like for example in this cartoon, so we just have 3 numbers here and we have 2 numbers here. So the third value was not quantified. And so if we leave it as an A, it means that we just ignore it. And so now the mean of the blue is closer to the mean of the red, meaning we underestimate the difference between the groups. Now we can also impute with 0, right? And so now if we consider the mean of the blue versus the mean of the red, suddenly the difference is much larger because this blue is an outlier, right? Because we set it to 0 for, lack of better ideas. So what MSSTATS is doing is something in between. So MSSTATS says, okay, we don't know what this value is. It appears missing. We assume that it's missing for reasons of low abundance. So let's try to infer the actual value which is missing. So what do we know about this? Well, first of all, we know that it comes from this peptide. And in this run, this peptide is below the observed peptides. Okay, so we expect this value to be lower. And in this group for these peptides, we see that there was a decrease. So essentially, we just project the same amount of regulation for the missing value. So this type of imputation requires that we have at least one non-missing value of other features in the same protein. And then this particular feature is quantified in at least one other run. So by learning the difference between the features from this group and the difference in regulation from this group, we can project that the missing value was actually this as opposed to this. And this gives us a better estimate of the difference between the groups. So here it underestimates, here is overestimates, and so here it is accurate if the assumptions are correct. So we do assume that these features, they're all parallel, there are no interferences. It's not always the case, but it is better than doing this. And it is also better than doing nothing, because doing nothing is actually not innocent, right? So now it overestimates the abundance in the blue group as compared to the true value. So this is what MSSTATS is doing. And so there's actually a fairly complex linear mixed effects model, which is associated with that. But in practice, what we do, we partition this into separate steps. So the first step only focuses on technology, so on the yellow part of this data structure. And we summarize this structure to predict what these censored values are. And we also do it in a way that is robust to outliers. Here, I didn't show this, but this summary is done in a way which is also resistant to outliers. And after that, we summarize all the features in the run in a single value per protein per run. And now we can focus on the actual experimental design, if it's time course or paired design and so on, and model it correctly. And this is a much simpler data structure with much fewer missing values than the original data structure that we had here. So this is what MSSTATS is doing. And so now, why is it useful? Well, so this is the IPRG data set with which you have been working for a while now. So this is one particular protein. So it's a spike in experiment, so we know the truth, right? So if we compare condition 1 and condition 2, so the true fold change here for this protein is 7.5. And so what we can see here is that if we consider the log of the sum, so this is a typical procedure that we can use by taking the sum of the intensities on the original scale and then take the log. It corresponds to this green summarization. It is so first of all, the gray lines, so this is the original values that we try to summarize, right? So we see here there's one peptide with high intensity, which is apparently an outlier. And so the green line takes the sum of the values of all of those dots, gray dots on the original scale, takes the log. Because it's a sum, it will be above the gray lines, right? It takes the sum of everything. And we see that this outlier here really affects the log of the sum. So the true fold change is 7.5. Log sum says it's 1.5. On the other hand, the linear model, which is essentially a glorified mean, will be affected by outliers, not only high intensity, but also low intensity. And it dramatically overestimates. So this is the yellow summary. The true fold change is 7, and it says 29. TMP stands for Tukey median polish. This is a robust summarization without imputation. It is doing better. So this is the orange summary here, but still a little bit off. And then MSSTATS does robust summarization and imputation. So this is the cutoff below which we don't trust the values. We remove these values, we replace them with this prediction, as I described before. And so it is the closest to the true fold change. And so the reason why we think it matters is because we think that it does give us more accurate estimation of fold changes, but also it makes our conclusions less dependent on the data processing tool. And so here is the evidence of that. This is our IPRG data set again. It's spike in, so we know which changes are true changes. Here is the summarization with log of the sum of all the features, and here is a summarization with MSSTATS. And the Venn diagram shows how many proteins are found differentially abundant, regardless of which data processing tool we used. And we see that the overlap is higher as compared to log sum. This is a couple other controlled mixtures that we have. Also, the overlap is higher, and we see this very consistently. So we have not seen yet a data set where this particular comparison gives us a different scenario. So we think that essentially by interpreting the data more appropriately, it allows us to have conclusions which are less dependent on some of the decisions that data processing tools make upstream. And so finally, just very quickly to finish, the community resources that can be available beyond just R. When we were working with these projects, we had accumulated a lot of data which we used to benchmark different statistical methods. And we realized that a big problem for those who develop methods, but also try to understand what are the best practices, is the lack of publicly available data on the quantitative side. So of course, there are databases such as Massive and Pride, which store a lot of identification results, but there is much less available as far as annotations of which run comes from which condition, and as far as the actual analysis of this data, which can be examined, which can be reproduced, can be maybe changed, can be questioned. And so we established a collaboration with Massive, and we developed a repository called Massive Quant, which stores, in addition to raw files and identification results, also the reanalysis of the quantitative experiments. And this was the project led by Meena. So Massive Quant has now 46 public data sets with 188 reanalyses. And the reason why I'm talking about that is because all these data sets have R scripts or some other analysis types associated with this data. You can work with more than just IPRG dataset that we present here, but we also hope that you can submit your own data and your own analysis or just reanalysis of the existing data to this repository. So it is not designed for us to store the data, it is designed for anybody to use and anybody to contribute the data to this. And so the repository, for example, will look like that. So we have the identifier of a data set, and now the containers will allow us to store different versions. For example, here Skyline versus MaxQuant, and here we take Skyline, we process with top 3 features versus top 5 features, top 10 features. So all of these analyses can be stored, and the results can be explored in a reproducible manner. The question is why there is still an empty cell of feature 2 and run 6. So I think you're probably referring to this. So feature 2 and run 6, yes. So this is what I said earlier, that some of the features we leave as missing. So for example, Skyline uses NA only when it cannot reliably quantify a peak. For example, if the 2 peaks are present, but they overlap so much that you cannot report their intensity reliably. Or think of a scheduled SRM where the peak moved outside of the window. So you kind of see the peak, but you don't have enough information to actually quantify. So Skyline specifically uses NA to indicate those. And so in this case, it is not appropriate to assume that they're censored. So we leave those as missing. So because we think that they are random, so essentially we treat them like that. We will just ignore those values. But for the majority of tools that don't make this distinction, it is more appropriate to assume that they're missing for reasons of low abundance. So there has been some research about trying to infer from the data which case it is missing at random versus missing at low abundance. This is good work, but it usually comes at the price of limited numeric stability. And we found that these decisions give us the solutions that work most often on most data sets and are most numerically stable. Okay, so this is for Massive Quant, and so this is just how the structure works. So we encourage you to submit your data. And very last thing, so obviously it is very challenging for us to present a lot of work. So MSSTATS has actually a lot more functionalities and a lot of more fun things that I have time to present. And it's not enough time today to discuss all of that. So if you are interested in learning more, I hope you can join us every May. We offer a program which we call May Institute, which lasts normally 2 weeks here in Boston in person. And we have a fantastic group of instructors who speak on a variety of topics. So it's not just MSSTATS, not just R, although there's a lot of that, but we also have people who design experiments, people who design studies, who make presentations. So we plan to do this again next May online, and we will dedicate a lot more time to both R and MSSTATS and other tools. So watch this space, and we will be posting announcements probably around Christmas time. So I hope this will be of interest. And of course, I have to now acknowledge again our group members, especially Meena, who led the MSSTATS work, and now Mateusz from University of Wrocław, who is taking over as the lead developer of MSSTATS. So it would not be possible without their work. And again, just ahead of the schedule, I would like to thank all the teaching assistants who are joining us today. So this is what I have to say about MSSTATS for today. I think now we can move on to the hands-on part. Again, if you think that this is too much and it's going too fast, please go to your teaching assistants right away, work on the materials of the first 2 or 3 days, make the best use of the TA time. Right now, everything will be recorded. And I will be here during the break after the hands-on part if you have questions to me specifically.
Okay, so then I will start MSSTAT tutorial part. So can you see my screen?
Yes.
Olga, yes. So same as yesterday, we have a two source. One is the GitHub. So if you go back to the GitHub now, we have a day 4 folders. One is for MSSTAT and the other one is a Cardinal. And then other source is our studio, a cloud, and then here's the day 4 MSSTAT. Yes, I will post yesterday the answer for the yesterday's exercise. Yes, I will. And then the today's as well. So today we will start day 4 MSSTAT, and then here we have some files over there. So one is for this is I just opened this file, and then next one is for exercise. And then I already uploaded some data set. So today I will just use the Skyline output for DDA cases, the same as the IPRG data what we have worked before. But data set itself a little bit different than what we used the last couple of the days.
Meena, there's a question about the R version requirements for MSSTATS.
R version now 4.0. We need a R version for at least 4.0 version. Yeah. So yes, so now here for our Studio Cloud, I already installed the MSSTAT. So you just call the MSSTAT before you use the MSSTAT one. However, if you want to install MSSTAT, here is the code. So if you just remove the sharp one, and then you can run it. But I recommend not to do it right now because it will take some minutes. So then I will just introduce this another function today to call session info. Let's just start to run it. This one shows all the information in R. So for example, here, R Studio, we use the R 4.0.3 version, and then this platform, which is not much useful. However, they also show what kind of package you have, what kind of the package is loaded for this the project. So you can double check it. So for example, so now we have a small the information there, but let's call the MSSTAT library, right? And then if I check the session info again, now we have a little bit longer list. And then here they show them these are attached to MSSTAT, and then this last number show the version. So when we have some error, or if you need to report what version of the package you use in the manuscript or somewhere else, you can use this information. This is how you can get this information. And also, if you want to post something in the MSSTAT Google group to have a question or error message, this is also very useful information. Okay, so then let's check the get working directory again, just double check. So now the project, the cloud and project, here's our locations. So MSSTAT need two different type of the files. One is the quantification file from data processing tool. So for example, the Skyline, MaxQuant, OpenMS, that proteome discovery for DIA cases, Spectronaut, and OpenSWAT, and so on. So now we can support around 10 different processing tools. That is this file. Input is from the Skyline. So that if you want to get this output, please let me know. So basically from the Skyline, you can export, and there's option for MSSTAT input. So they just automatically generate the MSSTAT required input format. That is this file. So let's call our input file first. And then here, this is running. It took a little bit longer than yesterday though. Okay, so let's see. Here today, if you remember what we practiced yesterday, we used the IPRG. This is the same IPRG, but difference is, here we have a much more number of the row. That is because the yesterday one, we used the protein level quantification, which is already summarized in the protein level from the feature level or peptide level or fragment levels. But here is originally from the feature level, which is the peptide, the PSM level, the quantification. That's why we have a much more number of the rows. So let's look at how it looked like. So here, so this file includes the protein name and then peptide sequence and peptide modified sequence and precursor charge and fragment ion, product charge and isotope level type and the condition biological file name, which is MS1 information, and area, which is the intensity value for here, and some extra information very specialized for Skyline. So from this, we can make the real MSSTAT required format there. So before we move on, just to review what we practiced yesterday. So let's check how many proteins information in our input. So let's check it this one. So here I want to get the length. Length mean how many count of the unique information of the row Skyline protein column. So it shows, yeah, here we have a 3,097 proteins are there. Okay, and then here also same thing. What is the unique information of the file name, which is the MS1 information? Here we have a 12 information. So, and then here is the one column is very specific for Skyline called truncated, which is already linked to the what Olga mentioned about the random missing and the sensor missings. So the Skyline very uniquely, they can distinguish 0 value and NA value separately. So both are available there. And then MSSTAT handle 0 value is a sensor, which means under the threshold and then which will be imputed. But NA value is basically from this truncated information. So they just have a mark, this peak is truncated or not. The truncated means if we see the chromatography, they just want to measure the area, for the night shape, but somehow they just measure just half of them and then not the correct area calculated. They actually have this information. So if the area is not correctly measured or measured, we just call this as truncated peak, and then just recognize this value is just random missing, which we will not impute. So here you can see they have a true and false value. So I just want to count it. Yes, there are, most of them are false, which means, correctly measured, but 829, the peak is truncated. So we want to handle this is just random missing value. So again, this is a very specific Skyline. As I understand, none of the tool give this information. In the cases, just missing value, we just treat everything is just censored and then just impute. So, And then the second information what we need is really your input. So not from the data processing tools, we need the experiment design part. So in this case, for example, here we already put the condition and then biological replicate. The Skyline itself, they have an interface you can put the condition and replicate the information. However, not many tools have this the functionality. In that case, we need another file we commonly call annotations, like this. So basically, you have to make this the file. So definitely, we need the MS run information, which is exactly same as this from the input. And then we need the information for condition and biological information corresponding to each run. So just copy your run information, and then you can just add this information over here. If you have a really large scale data, it will be a little bit difficult, but there is a kind of another discussion. So, and then also case by case. So if you have a difficulty for generate this one, also let us know during the TA sessions. So we have this one. So we need to call import this information here. So here a note Skyline, right? So this information. So now we are ready for quantification and then annotation files. Then very first function from MSSTAT is a converter. Here this input is from the Skyline, so we have to use the Skyline to MSSTAT format functions, which means, for example, these converters handle very tool-specific information to make a nice MSSTAT required format, and then filling some missing information. So Skyline already very close to MSSTAT required format. However, different tool, they have a very different type. For example, Progenesis, for example, the Skyline, this is the long format, but Progenesis, they just make a wide format quantification data. And then PD also, they have a different type of the format and different type of the column. So for example, Skyline sequence group protein, sometimes they just call sequence, sometimes they call the modified sequence, sometimes they call peptide, quite different way, diverse way they just define the column of information. So that's why we actually spend really a lot of the time to unify this converter to make the MSSTAT format, and then also which information is most useful, and then what information we have to use, and so on. We actually spent a lot of the time. So I, personally also recommend you to use the tool-specific converter in MSSTAT, even though you can make your own converter, because sometimes that converter still can generate some error for the next functionality. Okay, so how many features are considered, one of the questions, how many features are considered good for the data set? I'm not quite understand the point. So number of the feature is really depending on the data. DIA definitely have a much more number of the features, and then DDA cases, we have a small number of the features. But if your question is how many features per protein we have to use it is also quite different. So I will talk about we have some also option how many features we can select it in the the summarization level. However, we also have another algorithm and statistical method to find the most informative feature from the data set. Olga, do you want to add comments?
Yeah, I can just add that a short answer is a few, but not too many. Right, so this is a very vague answer, but for example, in DIA, you can quantify up to like 100 features per protein. Chances are you don't need that many. It will complicate your computation a lot, and probably some of them are closer to the noise level, and they will probably harm rather than help. So essentially, a few good quality features go a longer way than many noisy features. And how you define this? Well, there are multiple ways. You can select the top that many most intense features. MSSTATS has actually a functionality which selects quote unquote informative features and sets aside others that you can either explore by hand. Maybe, for example, they have PTMs or something like that, or you can just discard. But I think the short answer is that one feature, often you don't know if it has interference or not. Too many features, it's not necessary, not necessarily productive, but you want to have a few good quality features per protein.
Okay, so I just run it because it will take some minutes, so it is already done. So if we use run, you can see it show the progress. So what actually this converters are doing is they just remove the some protein name is decoy, and then they just replace the truncate peak, replace with the NA. They just remove the some feature, have a very few value, which means 1 or 2 values are there. Or some features all NA or all 0, which is not that much meaningful for further analysis. So we just replace it and so on. So it shows the progress over here. If you want to know what kind of options it has, you can just make a question mark and skyline MSSTAT format. If you want to use other processing tools, so you can go to the MSSTAT.org, our webpage, go to the MSSTAT, and then here is our user manual. It showed pretty much all the different cases. So for example, here, analysis here, for each different processing tools, how you have to use it, what kind of option you have to change it. Each cases are addressed here. So please check this manual. So yeah, so then how this, the processed data look like. So now, here we go. So now we have a protein, peptide, precursor ion, fragment, product, and so on. So now we don't have any extra columns. So this is the standardized MSSTAT required format. So after the any processing tool converter, we will have the same number of the columns. Then we are ready to move on. Before we move on, I can just want to shortly check it. This is also commonly what I do before we move on. So for example, let's check the protein number of the proteins. So previously, what I remember is 3,097 proteins. Let's check how many proteins we have. Now we have a 3,027. So which means around the 70 proteins are somehow removed because they might have a lot of the missing value, so that's why they can filter it. Or some of the protein include a decoy in the protein name. These proteins are also removed. Then also commonly what I also double check is always checking the missing values. So here these cases there are, I just want to count, this is a count a missing value, which means from input a skyline data in intensity column, is there any NA value, and then count it. So there are 219 missing values there. And then second one, this one is, I want to count how many 0 intensity value they have. So first is what I count is this, this mark is not. So means intensity value is not in NA value, and intensity value equal to 0. Then it shows there are more than 2,000 0 value, 0 intensity, which we're going to impute it later. Then this is the second step. No, this is actually the third step. The first one we have to, so here's the first. So first we have to install the MSSTAT. Second, we have to import the data. And next, we just need to use the converter to make a MSSTAT required format. And then let's do the data processing, which includes the normalization and then imputation and summarization and so on, what the Olga says. So here's the function called the data processing function. This is a quite unique function for MSSTAT. So here, the raw file is input skyline, which is output of the Skyline MSSTAT, Skyline to MSSTAT converter. And then we want to do the log transformation for intensity. And then we want median equalized normalizations. And then we want to summarize the Vitek median polish. And then we want to impute it. And then the sensor int means which value we are going to impute it. So there are two options. So one is a 0 or NA. 0 or NA. Right? So if you put the NA means we are going to impute the NA values. If you put the 0, we are going to impute the 0 value. So this is also different by tools. For example, Skyline, we have to put the 0. If you change this one to NA, the conclusion might be different. For example, max quant cases, we have to put the NA because the max quant generate the missing value as NA. Progenesis case, we have to put the 0 because the progenesis generated missing value is a 0 and so on. So this is a very tool specific. If you are not sure, you have to check the manuals. This is the old default, which means if you just remove and then just put the input and run, that is the default. You don't need to assign it. They're gonna impute 0 value and then summarized by Vitek median and so on. But I just want to make sure we actually used the options. So let's run. Then what you're gonna show, this function is the most time consuming the functions. So for example, here, they just show them, okay, there are this much number of the intensity is the 0 values. And then we are going to replace this one. That is replace 1. That is because, we are going to do the log transformations, but log 0 is, it doesn't work. So we just put the log 1, which means just still 0, 1. So that is kind of the, the tricky part. And then here's also under the cutoff means MSSTAT itself, MSSTAT also defined some new cutoff for sensor, which means a limit of the quantification. In this case, this is a 16 something. So under this value, we're gonna see this value should be imputed. And then MSSTAT just summarized how MSSTAT recognized the experiment design and then what kind of information they have. So for example, here, number of the protein, we have more than 3,000 proteins are there. And then also summary here means number of the peptide per the proteins, how many they have. So which means some protein, they have a 2 peptide up to 196. And then here also show the number of the transitions. So this case is a DDA, so it should be 1. But DIA cases or SRM cases, you can have more than 1 values. And then this is also very important part because MSSTAT inside, even though we didn't tell that this experiment design is group comparison design or time course design or pair design, we didn't mention it. But based on the annotation information, MSSTAT recognized this design and then apply the best model for this experiment design. So here's how they recognize it. So for example, here for condition 1, there are 3 MS run, and then there are 1 unique biological replicate, and then they have a 3 technical replicate and so on. So this is how they recognize it. If you see, and this number is not matching, you have to go back to the annotation file, and then you need to double check, is there any kind of misinformation or not? And then if they just list of some potential issue cases, so for example, some transition are completely missing and needs the 1 condition, which means, we have a 12 run, but some feature, this feature actually completely missing, for example, condition 1 and so on. So this is a kind of the just show how we recognize the data set. And then, oh, this is already done, but this is actually show the progress. So start from the summarized per subplot, which means we are summarized, the protein level summarized per protein per on the run, and then this 100 is done. Here, when you see the some people is kind of, oh, what's going on? Is this error message? It's actually not an error message. This is a warning message. So you don't need to worry about too much about it. Like for example, here we already have another warning message here, warning message. So you don't need to worry about it. If you have an error message, they technically say error and something. So this is the data processing. Then let's see what data processing output has it. Let's just see. Right, so there's a question, how can I know what other normalization method you can use? So so data processing function, if you question mark and data process, this one is a help file and they show all the options, what options they have. So for example, normalization, I just put the equalize the median, but we support 4 different normalization methods. The one is a no normalization. The second one is, which is default, is equalized median method, make a median of whole log to intensity per run become the same. And then other thing is the quantile normalizations. And then other thing in the last one is a global standard normalization, which means we know which peptide or proteins are standard, and then which means it should be the same amount across the runs, and then we use the specific peptide or protein should be used. Then how you know which one is suitable and the appropriate way to do it, it is really depending on your data acquisition type and experiment design. So for example, let's say DDA, DIA cases, if the number of the protein quantifies more than 1,000 cases, we commonly recommend just an equalized median method. For SRM cases, more tricky. So if label-based SRM cases, we also recommend the equalized median one. But label-free SRM, the more targeted approach, but without any reference and a small number of the proteins, equalized median method is risky. So it's really depending on the situation, but I do not recommend it. In the cases, if we recommend to use some standard or reference of the peptide to use it. To use this, actually, you need to consider this one before you actually set up the experiment. So at the beginning, before, during the, sample preparation level, you need to consider this normalization method already. And then you also need to some spike in protein over there. And then we have to use this normalization one. If you have a very specific experiment type and so on, and then not sure the normalization, please ask a question, during the TA sessions. So, right. And then, okay, so data processing. So the quant skyline include all the output. So let's see here, quant output. So as what we practiced yesterday, other things look like the table. So they have an observation and variable, but quant skyline, it looks a different type. So the least one, which means they have a 5 different elements, and then each one have a something. So how we know, as what we practiced yesterday, names, quant, skyline. This is how we practice when we try the t-test. They have a different information in the different format, different the data type format, but they just combine everything and then just save in the one space. So here's how the quant skyline as well. So for example, skyline, quant skyline, which is output of the data processing, have these 5 different elements. So this first 2 is the most important one. So let's see the quant dollar and then process data is the data frame. And then this is how it looks. So this is the very similar as the input guideline, which is the MSSTATS required format, but they have some extra columns and extra information. So for example, here, intensity is original intensity, but this abundance is log transformed and normalized abundance, which we will use for next step. So the intensity is the original intensity, but abundance is somehow already processed out of values. And then the fraction, even though this case we don't have a fraction, they also recognize the fraction information and then add this information over here. And then the censored, they also mark which value is censored or which value is not censored, and so on. So this is still the feature level data, but add more column and then more informations. Then the next important one is a run level data. So my logic how to name is it's just data, but already somehow process the data like the log transform and the normalization. And then run level data is a run level summarized data included. So let's just see. This is the run level data. So this is, it means for run one, for this specific protein, summarized value is this. To summarize this value, we used 8 features, but there is no missing values. And then there's no features are imputed. If you can search in some cases, definitely you can see number of the features 8, and then maybe missing percentage is a 50% or something. And then it showed how many the features are imputed, like 3 or 4. It doesn't need to be same as the exactly the missing feature, because if they have a random missing and sensor missing together, random missing will be not imputed. So this one is only show the censored of the missing value, which will be the imputed. So this is the how the run summarization. So if you want to go further for something else, just use the MSSTATS for just the protein level summarization. This output is the what you have to use it. And then the yesterday what we practiced, which is the protein summarization, is this data set, not the process, not from the processing tools. Okay, so after we do the summarize, we can make a visualization. We want to see the whole output, how it look like. So in that case, we can use the function called data process plot. They support 3 different type of the plot. One is a QC plot. Second one is we call the profile plot, and then the other one is a condition plot. So for this function and then parentheses, we actually need to say which data set use, which is output of the data processing. And then we have to call what type of the visualization you want. So here I just call QC. And then width and height is the PDF size. And then which protein we use. If you do not define this, they're gonna generate for every single protein. So now we have more than 3,000. So you're gonna generate more than 3,000 plot in the one PDF. But for example, QC plot, I just want to see just the first one with the all proteins. So in the cases, I call which protein, all only. And then we have to give them some file name. So in that case, I can do this, but in that case, I will just remove this one, and then I will run, right? And then now under our folder, now we have the PDF file. So click it. This is the we call QC plot, the box plot, to see all the distribution of each one. So now after our quantile normalization, equalized median normalization, this is show the median value. It should be the really equals. if we use no normalization method, you can see some variation. Some, this bar is up and down and up and down. Right, here I just this a quick no, this is not just, you have to remove this part, output. So this is actually define the folders. So either way, you can just remove this output, or you can make the new folder output. Then all the file will be saved in the output here. Yeah. So, so QC plot is very useful to see the normalization, or sometimes, not that much of it, sometimes some run is really high or really low, is a really outlier. And then cases we can double check it. So just, quality checking, this QC plot is also very useful. Next one, which is my favorite one, is the profile plot. Same function, data profile, but type is the profile, so not the QC, not the QC. Okay, first one, I do not recommend to run right now, just to keep it as the later. Yeah, keep it later. Yes, so Meena, just remove the output / part. Just remove the output / part. So yeah, this one, just to keep it, because it will gonna generate more than 3,000 proteins plot, and then it will take a while. So let's try this second one. Exactly same, but I just say I want this specific proteins. I want to draw the profile plot for this specific protein. So, okay, here you can remove it. I will remove this one and then just try run. Right, now we have a two plot. One is the profile plot, and then the other one is a profile with summarization. So I will click the profile plot one. This is the visualization for our feature level data. So here, for example, here we have a 1, 2, 3, 4 condition, and then each condition we have a 1, 2, 3, each 3 MS run. Each line means each feature. So in this case, it's just a peptide charge. And then each dot is each feature intensity, So now, why this is kind of important is just to see, is there any kind of features that have some issues or something really weird pattern or something like this. So for example, here, quite a parallel across the feature, but the specific feature is kind of up and down, and they quite behave differently. In the cases, you might go back to your processing tool and then double check what's going on. Maybe there are some parameters you can just change it, or you can remove this one, and so on. Just want to give you some idea about the data set itself. Other one is with the summarization. So let's click this. So difference is now we have a gray one, which is the feature level, process the feature level data, which is exactly same as this, just change the color. However, we just overlay, the protein level, summarize the value here. This is by MSSTAT, after imputation, after the Tukey medium summarization, which is also from this run level data. So again, gray line is from the processed data, and then this red line from the run level data. And then for next step is the hypothesis testing. We are gonna use this run level data. We are not using processed data anymore. So this is how we can just draw the one protein. But if you just remove this, I do not recommend to run right now. After you can just run during the TA session. They're gonna generate the one PDF file including all proteins profile plot. And then another thing is the third one is we call the condition plot. So here also let's run this one. Here. So let's remove the output. right? The condition plot is kind of similar as what we practiced yesterday. So for each protein, this plot used the feature level data, not the protein level data. Just calculate the mean value and then calculate the confidence interval, and then just draw it. Just give you some brief idea how the data look like. Okay, so this is how we have to do the data processing more. But I think we don't need to run this part, but just in case, so what the other options make different conclusions. So for example, here, always MSSTAT default is always do the imputation and the summarization. But if you don't want to impute this function, you have to change it. So MB means model-based imputation. Previously, the default is a true, but if you don't want imputation, just change this false. Other options should be same. And then the next step, so just remind again, first read road MSSTAT. Second, we need to import the quantification data. Then just use the tool-specific converter. And then we do the data processing to get the protein level quantification data. Then we are gonna do the inference hypothesis testing. So to hypothesis testing, definitely we will use output of so there's a question about the max this one. This is how we define the MSSTAT specific censored cutoff we defined. So which means, if you remember the Olga's presentation here, how we defined this cutoff is this option, the max quantile for censored 0.999 is how we define this line, this dashed line. So that is how we calculate it. So this is from, we draw the distribution of the log intensity and then calculate that this 0.99 percentile one, and then make a completely opposite way. And then here's our cutoff. So, but if you want a more conservative way, which means close to the center, you can make a smaller one, which means close to the center. If you want to more wider, you can make much bigger number, close to 1 value. Or if you said NER, they do not define this line in MSSTAT. They just use the original 0 or the NA value from the processing tools. Right. So then the last part, the hypothesis testing part, we have to say which comparison we want to MSSTAT. So they know they means the MSSTAT know we have 4 different the condition, but we have to tell which comparison we want. So to define, this is the tricky part. This is always very important. How the MSSTAT recognize is group is from the process data set. So first, always we need to double check how they just recognize this group, and especially order. So now they recognize the condition 1 is the first condition, and then second condition, third condition, and the fourth condition. They just recognize the order of the condition in this way. So this case is quite easy because 1, 2, 3, 4 is quite easy to commonly we can see the order. However, sometimes if we say healthy disease, MSSTAT or R just define this order by alphabetically. So disease is probably first and healthy. But if you think healthy is first and disease, this order is really matter how you define the comparison. For example, healthy minus disease, disease minus healthy. In that cases, the log for changes calculation will be completely opposite, like plus or minus cases. So I always just recommend to check this order. And then based this order, you have to define which comparison you want. Let's say I want to compare condition 2 minus condition 1. In that case, what we have to do it, first, let's see, first to make this matrix, but now we have a for condition, right? And in this parenthesis, make a for space. And then the first space for condition 1, second space is for condition 2, third space is for condition 3, and the last space is for the last condition 4, right? And then we have to define the constant. So for example, condition 2 minus condition 1 is the same as condition 1 minus 1 times condition 1 plus 1 times condition 2, right? And then if you want to add, actually we don't need the condition 3 and condition 4, right? In the case, 0 times condition 3 and then 0 times condition 4, right? This is the same as condition 2 minus condition 1. And then we just put this constant in each space. So for example, first place is for condition 1, so we have to put the minus 1. Second space is for condition 2, we need to put the 1. And then third one, condition 3, we don't need it. And then condition 4, we also don't need it. This is for condition 2 and condition 1. Let's practice one more thing. Let's say I want to compare condition 2 minus condition 4, right? This case, we don't need the first condition, 0 times condition 1, but we need the condition 2, 1 times condition 2, and then we don't need condition 3, 0 times condition 3, and then the condition 4 should be minus 1, minus 1 times condition 4. Also same thing here. I just add the first one, condition 1 position is 0, and then 1, and then next one is 0, and then the other one is the minus 1. Right? So we can do like, we can just set up this contrast matrix is this way for pair comparison. The good thing is MSSTAT also can compare some linear combination. So this is the advanced one. I hope everyone is just follow me. Let's say I want to compare mean of condition 1 and 2 and condition 1 and 2 versus mean of condition 3, condition 4, right? Let's write this one is an equation. So to calculate the mean 1 and mean 2, what should I do is condition 1 plus condition 2, and then we have to divide by 2, right? To calculate the mean, right? And then minus to calculate the mean of the other two, C3 plus C4. We have to combine these two and then divide by 2, right? Then I just want to spread this out, spread this formula. How we can call, so 0.5 times condition 1 plus 0.5 times condition 2 is this 2 number is mean of first two condition, and then next one is 0.5 times C3 plus 0.5 times C4 right? But we have a minus here. So I want to remove this parenthesis, but we need to change this one is minus, right? Then we can write this comparison matrix. So for example, in the case, just first projection is a 0.5, second is 0.5, third one is minus 0.5, the other one is 0.5. So if you set up the metrics like this way, you can compare mean of the first two condition versus a mean of the second, condition 3 and condition 4. So this is how we define the matrix one. So in that case, you don't need to worry about it. I just already set up for all pairwise comparison. So for example, here I want to compare condition 2 minus condition 1. And then this second one is condition 3 versus condition 1. The other one is condition 4 and condition 1 and so on. So here is the my comparison matrix I want to compare. Then this is also the second major function called group comparison. So we have to give the two input. One is which comparison do you want? So you have to give the contrast matrix, and then data, which is output of the data processing tool, data processing functions. So let's run. Then not much things because MS they recognize experiment design and then already find the best model for each proteins. And then it just show the progress so far here. Everyone okay so far? I think we have a couple of seconds. For timing wise, DDA cases, just few minutes. I think so total, if you already have a defined script, it will take off like 5 minutes for one DDA case. DIA cases also is really depending on number of the protein and especially number of the MS run. Around 20, 30 MS run. And then number of the protein is kind of around the 5,000. For my laptop, just regular Mac Pro, it will take like 10 to 15 minutes. But if you have a much bigger data set, it's actually take a really while. And then we actually trying to refactoring the code and then just try to solve the memory issues. Any questions? We are almost there. Right. And then here's a test skyline, which is the output of the group comparison. This is also list, and then they have a 3 element. The important, if you want to see what information they have, there's a skyline, test a skyline, and then we have a 3-1. Comparison result is also probably most meaningful you. So let's see. Here I just get this comparison result matrix and then save is as the skyline result. Let's run. And then let's see how it looks like, skyline result. It has all the testing result. So for example, here, this protein compare between condition 2 and 1. This is the estimate log for changes, and this is the standard error, and then degree of freedom, p-value, and adjust p-value, and some issues. Issues column means if I want to compare condition 2 and condition 1, but condition 1 is completely missing, which means we cannot compare. In the cases, issue said one condition completely missing. However, they have a NA value for P value and something. However, they just put the 0, 0 in the adjusted P value. That is the skyline result. So if you want to save this one, there are two different ways. Definitely you can save this file as a R data type, or you can save this as a CSV file. So in the case, let's remove this output folder. Then let's save it. Then now we have a CSV file. So you can use this information wherever you want, or you can just submit this result somewhere else. Just for advanced people, as I mentioned, there's also another fitted model. So which show all the fitted model for each protein. So that's why we have a really quite a big size of the file. So at some point, maybe if you need, we can just come back to this fitted model and then just get the more information over there. We are getting close. Okay, so this is the skyline result. Okay, I know, but I want to see only the significant protein or significant comparison. So in that case, we can use what we practiced the last couple of days. Here I use, from the skyline result, the square bracket, and then comma, and then I want to get the rows include how just p-value is less than 0.05, right? And then I just save this as a significant proteins. Then let's check it. The n row means count how many n number of row. It means number of the rows. There are 29. And then let's see significant proteins. It shows the row with just p-value is less than 0.05. So for example, here, this protein for the specific comparison, this much, and then this is the just p-value. So you can get the subset of the significant proteins only. Or more, you want to get the subset of the proteins significant and the estimate log for changes itself is greater than 2. Then you can add this one. And then last part, as we practiced yesterday, I want to make a volcano plot for this comparison. So as we practice, we can also draw the customized volcano plot by yourself. However, MSSTATS itself, they already defined a function called group comparison plot. So the data should be output of the group comparison, which is a skyline result. And then type, I want to draw the volcano plot, and then you can define your file name, right? Just remove the output, and then here. So I just click this one, and then this is the volcano plot. So this means for condition 2, condition 1, this is the volcano plot. And then this line means just a p-value cutoff 0.05. And then this is no regulated. And then this part is down regulated and up regulated. In this comparison, we defined the 6 different comparison. So this is the volcano plot for the other comparison between the condition 3 versus condition 1, and so on. So this is the part how you automatically can generate the volcano plot. Yeah, so here's also another option. You can just change the sum option and then make a different cutoff, and then differently you can show it. Last part, the MSSTATS, the last function is calculate the power and then calculate the sample size. So here we call the data sample size function. In that case, we have to give them the fitted model. So this is from the test skyline. We have to give the fitted model there. And then give them the desired for changes. And then I want to calculate the power if I have 3 samples per condition. And this is how they just report. For example, if your desire for changes is 1.2, the power is 3%. But if your desired for change is 1.5, the power is 97%, and so on. If you want to calculate the sample size, you can just change this option. So now in this case, it's same function, but number of the sample true means I want to calculate this one, but we have to give them the estimated power, and then it shows the number of the sample. So for example, I want to have the 90%, but desire for change is 1.2. In the cases, you need 11 sample per group, and so on. So we already over time, so sorry for all the delay, but just play around with the TA. But I will give you the, Melanie, is it okay? You can start at 2. I just want to give them some 15 minutes plus some break. Is it okay?
Melanie is not there, but I think we can say
yeah,
certainly 2 or even like past sometime past 2.
Right. Yeah. So that you can definitely, you can play and then draw the other plot, or if you want to exercise more, here's the example. So here's the exercise. This is the case for max quant. The data set is already in our cloud. So here I just make how you import the data set, but I just leave some space so you can try the MSSTAT. So please try this exercise with the TA, and then if you have more questions, let me know. The MSSTAT website is, very simple. MSSTAT.org. MSSTAT.org is our website, and then it shows all the package as well. Okay, so thank you. I will, yeah.
So what time do we come back?
2, let's do 2.
Maybe 2:05 to give enough time for everyone. Okay, let's say 2:05. So now it's 1:43 Eastern, so we have 20 minutes. Please visit your TAs if you have questions, especially if you have some error messages or something like that. And we will be back. And the second part will be not about MSSTATS anymore. It will be about Cardinal, which is a R software for mass spectrometry imaging. And so we hope that you will also see how R can be used for these other types of applications, especially applications that require reading raw data. But at the same time, if this is not something that is of interest to you, we do encourage you to go to TA rooms and spend more time with them and ask questions. Please don't be shy. You can ask other questions, not just about the exercises. So do take advantage of the TA time that we have. This is a very rare opportunity.
And plus, the cardinal part, I will be one of the TA. So if you have more question, I will post my TA link over there. Please come to the link. Yeah.
So during the Cardinal, if you would rather work more with MSSTATS, you can just join Meena. Okay, thank you very much, and we will see you in 20 minutes. Okay, so we are now about to start the second part, which focuses on mass spectrometry imaging with cardinal. Just wait a couple more minutes. Okay, here we go. So it is my pleasure to introduce Melanie Foell, who has a joint appointment at University of Freiburg in Germany and at Northeastern University. And so Melanie is an expert in cardinal. And so I'm looking forward to hearing from Melanie about the use of the package.
Hello. So we will spend the remaining time of this course on mass spectrometry imaging, which is a very specialized but also very exciting mass spectrometry based technique. I hope you can see my screen.
Yes, we can. All is good.
Yeah, but I can't. Oh, sorry. So I can't move my windows. So you should already have the link to the RStudio Cloud. And I would also like to share with you the Cardinal link for the Cardinal website. I will put it into the chat. Here you will find a lot of resources like data sets and many user guides and so on. So I really recommend to visit this website. And there's also a vignette out of which I will cover a part today. So for today, I have a presentation about mass spec imaging, data analysis, and cardinal. And then we have time for hands-on analysis of a real data set. So let's start with the theoretical part. So mass spectrometry imaging is abbreviated as MSI, and it enables the label-free measurement of spatial distributions of many different molecules. For example, metabolites, lipids, peptides, proteins can be measured in complex biological samples for their spatial abundance. Here I have depicted some examples. So on the left, you see a mouse bladder cross-section. And mass spectrometry imaging enables to dissect the different tissue histologies. In yellow, the outer line is the adventitia. In blue, we have the muscle tissue. And in pink, this is the urothelium. But MSI is not only able to distinguish different tissue histologies, it also enables to find heterogeneity inside tissues where you would not immediately see it in a visual inspection. On the left side below this bladder, we have an example of the different molecular composition of liver metastasis. So they show different molecular composition depending on the position inside the metastasis. Biomarker discovery is a very common application of mass spectrometry imaging. Here's an example of tumor and normal renal cancer tissues. And due to their molecular different composition, we can build a classifier that later on would enable us to classify new tissues in either tumor or the normal group. And this is, of course, very promising for clinical applications to find potential diagnostic biomarkers or also markers that predict therapy outcome. MSI is also used in the pharmaceutical industry and in pharmacological research to image the distribution of drug in study animals. So the first image shows the spatial distribution of a drug inside a rat sagittal section. And the next two images show that the metabolite is distributed to different organs than the drug. And this is, of course, very helpful for pharmacokinetic and toxicologic studies. So how does this technique work? What's the technology behind to generate such nice molecular images? I will show the data acquisition based on multi-TOF mass spectrometer. And this is the most common type of mass spectrometer used for MSI, but there's many other mass spectrometers and different ionization sources and mass analyzers that work for MSI. The principle is always relatively similar. So we start with a sample that's often a thin tissue section that's mounted onto a glass slide. The glass slide is quite similar to a normal microscopy glass slide. Sample preparation steps might happen. Different steps are necessary for different types of samples, different analytes, or different mass spectrometers. So for MALDI, for example, we need to spray a matrix on top of the tissue. So this tissue then on the glass slide is inserted into the mass spectrometer, where the MALDI laser scans over the tissue with a spatial resolution in the low micrometer range. And this means that in thousands of tissue locations, the laser is ablating the surface of the tissue. The peptides or molecules that we want to measure get ionized, and their mass is measured in the time of flight analyzer. In the end, for each position where we shoot onto with the laser, we obtain one full mass spectrum. So MSI is normally only done on MS1 level, and we generate thousands of such spectra. Later on, we can still stain the tissue, which is quite helpful to compare the optical image of the tissue with our molecular images. The molecular so-called ion images we obtain by plotting the intensity for a certain mass, which should correspond to one molecule, in all the spectra that we have acquired. And then, of course, we will see that different ions have a different distribution in our tissues. So this is an example of a pig fetus, and the left image is a molecule that's more abundant in the heart. And here on the right side, there's an ion that is more abundant in the liver. So another representation would be this 3D data cube. We can think of the data as having an X and Y coordinate, what would be the spatial position of each spectra or pixel. So I might use the terms interchangeably because in one location or pixel, we acquire a mass spectrum. And then we can think as the molecule intensity maps as the third dimension, where we have many layers. And depending on which layer we look at, we see that there were different intensities in the different pixel locations. What is interesting here is that we have in all dimensions, we have like tens to thousands of measurements. So we measure several 10,000 of spectra, and we also measure several 10,000s of MC values. So in the end, we get quite a large and complex data set. And with all the advances in this rapidly changing technology, we get better and better spatial and mass resolution, leading to even more measurement points and even bigger data files. And single data files can be several hundred of gigabytes in size. Another thing that makes the analysis quite challenging is that we have dependencies in both directions. So we have mass features that behave similarly. For example, all the peptides of one protein or fragmented ions or adducts. So there is some dependency in the mass direction, but also in the pixel direction, because the pixels are spatially related. Pixels that are next to each other have a similar molecular content. And so also we have a spatial a dependence in the spatial dimension. And to analyze this quite distinct type of data, you need specialized software. And I would like to introduce you to the Cardinal software. The Cardinal software is a free open source R-based software that is distributed via Bioconductor and was already downloaded more than 7,000 times since its release in 2015. It also won the John Chambers Statistical Software Award, and Kylie Bemis is the lead developer in the Vitek lab, and she developed not only this general software, but also very specialized and powerful statistical methods. And furthermore, she developed the Meta package, which is also a bioconductor package, which enables working with larger than memory data sets. And that's quite important because, as I just said, the data sets become bigger and bigger. So this is quite nice. And Cardinal also comes with an extensive documentation and has an active mailing list. So here is an overview of the many Cardinal functionalities, and they are grouped by common MSI analysis steps. In reality, an MSI data analysis is not so linear. It's a bit more repeatable, but this will serve to explain some of the steps and functions. So Cardinal is based on the IMCML file. MSI raw data is commonly stored in this format because it's the open standard format, and each vendor software has now an export option to directly export such IMCML files. As you have learned by now, of course, there's also many other file types that can be imported into R and included into the data analysis. Cardinal comes with plenty of options for visualizing ion images, mass spectra, and statistical results. There's also preparation steps that can be done within Cardinal, such as annotating regions of interest, subsetting in all dimensions, such as subsetting and filtering masses, spectra, and intensities. What is really important is that we can combine several files inside Cardinal. And this allows us to use Cardinal not only for the analysis of single files, but for complete MSI experiments. Preprocessing is quite an important step to remove noises and shifts. And here Cardinal has such a big portfolio of methods and parameters that it can handle a lot of different MSI data. With the cleaned up preprocessed data, we are able to perform statistical analysis. And here Cardinal really stands out by providing not only basic statistical methods such as colocalization and summary statistics, but also very dedicated and powerful algorithms for classification and segmentation. Last, we can export many things out of R, as you have learned of today. And what really makes a difference here is that from R, we can export our code and our markdown files. And that means that we are able to share the code that we have used for the analysis with our colleagues, but also with the whole community. And in many MSI softwares are either proprietary or the analysis is based on in-house scripts that never get published. So with Cardinal, we are really able to bring forward an MSI data analysis that allows a transparent and reproducible data analysis. And this works even with parallel CPU computation to speed up the analysis time for large data set, and also with out-of-memory computation, so that it's possible to analyze data sets that are larger than the computer's memory. So now some examples. So here are some visualization options in red. This is the code that is enabling such visualizations in Cardinal. We can plot mass spectra, either single mass spectra, average mass spectra, zoomed in mass spectra, and we can also overlay some mass spectra. And then each spectrum can be assigned with different color. In terms of ion images, these are these nice heat maps that are so popular. We can either plot just the raw intensities, or we can also enhance the contrast or smooth the images a bit. All these functions will only change the image. They will not change the underlying data. And last, we can also overlay several mass features into one image. And this is again the mouse platter section. And here we have overlaid 3 masses that are distributed in distinct morphologies. Here are some examples for pre-processing steps, smoothing and baseline removal, and peak detection. So smoothing is necessary to remove some noises in the spectrum and make the peaks really nice and smooth. Baseline removal is quite a typical step for MALDI data, and peak detection, yeah, you anyway know what it means. And this is also an important step for any type of MSI data. Just as a reminder, before the peak detection, we have profile spectra where we have several data measurement points that describe one peak. And afterwards, we have centroided spectra where for each mass, we only have one intensity value. So for statistics and statistical modeling, there are 3 main analysis types in MSI. The first one is class discovery. This is normally done by unsupervised segmentation. That means we have no prior information of the data set, and we generate classes that group together tissues with similar molecular content. And for this, Cardinal has many options, and the 2 highlights are the spatial shrunken centroids algorithm that works as a multivariate segmentation, meaning it uses all MC values, and the spatial DGMM is quite special in the field as well because it operates on single ions. Then if we have data sets where we know our classes and we want to know which molecules are different in the different classes, we can just use a class comparison based on linear models. And for this, there's the means test, where we just compare the means between the different groups. And there's also a segmentation test where we previously do a segmentation and then only compare the mean intensities of the segments, what might be quite more accurate than the mean of the whole sample. And third, we could do class prediction via a classification analysis. And here again, we have tumor and normal tissues, meaning we have some labels or classes, and we can now train a classifier to learn the molecular content of the different classes. And when provided with a new tissue of an unknown class, the algorithm could potentially predict if it is a tumor or a normal tissue. And for this, again, we can use the spatial shrunken centroids algorithm in a supervised manner by providing the class labels for each spectrum. And last, Cardinal is based on the IMCML file. It can read it and write it. And the IMCML file, as I said, is the open standard file format. There's also a converter available and a validator to make sure that the exported or converted IMCML file is valid. The IMCML file consists of 2 parts. The first part is also called IMCML. This is file number 1. And in this file, we have an XML-based human readable file that contains the metadata, such as the position of the mass spectra in the tissue. And the second file is an IBD file. This is only machine readable. And inside we have the different mass spectra. Inside the IBD file, the mass spectra can be stored in 2 different ways. We can distinguish a continuous file type, In this, the MC array is only stored once. And in contrast, in the processed file type, for each spectrum, we have not only an intensity array, but also an individual MC array. And Cardinal can handle both data types. And during the course of an analysis, often the types also change. And we will see this later. And don't get confused. So even though this is called a processed IMCML file, this has nothing to do with pre-processing or any type of processing. It's just how the file is, how the file type is called when it stores the MC array for each spectrum. So today we will use this pig fetus section as a data set for the hands-on part. You have already seen it before. It was imaged with a DAISY-LTQ mass spectrometer in the MC range from 150 to 1000. And what we will do is we will load the data, explore the data, pre-process the data, segment the data, and finally visualize some interesting ions that hopefully look similar than the images that we see here, where we have distributions that correspond to the heart region or to the brain or to the liver region. As I said, what we are doing today is part of the vignette, which is available also from the Cardinal homepage I sent previously. So what are we going to do now? First, we load and explore the MSI data. So normally we have an IMCML file that we load with read MSI data or read IMCML functions. But today we will use a data set out of the Cardinal Workflows package, so we can easily load it directly from the package. And we just need to be careful because this is still stored in an old format. So we will coerce it into the current MS imaging experiment type of object. So again, this is one type of representation in 3D. But actually, if we annotate the spectra only by metadata with their X and Y coordinates, we can also have a 2D representation. And this is more how the raw data looks like if we extract it from the objects in Cardinal. So each row corresponds to a mass feature and each column corresponds to a spectrum. And this means that if we look at the intensities of one column, we obtain a mass spectrum. And if we assemble the intensities of one row into the right X and Y dimension, then we obtain one heat map. So we have three main types of raw data. And to extract them and maybe also manipulate them, there are many different functions which we will look at. Next, we perform pre-processing. This is the pre-processing workflow that we will do today. We start with the raw data and we calculate a mean spectrum. On the mean spectrum, we perform peak picking, peak alignment. This means that small mass shifts are aligned together. And we perform peak filtering to obtain a mass list. And then there's like a second branch of the analysis. We start again with the raw data. We normalize the intensities. And now comes a really powerful peak binning algorithm. And what this algorithm does is depicted on the right-hand side. It takes the mass list that we have obtained via peak picking, peak alignment, and peak filtering as a common mass list for this data set. And it peak binning looks into each mass spectrum at the positions of the MC values that are in the mass list and applies a tolerance that we specified to them and looks where inside this window is a peak. And if there is a peak, the area under the curve is integrated and used as an intensity value. So after peak binning, we have again a centrated data set. And the advantage is that we are not so dependent on how good the peak picking worked. And we have way less zeros in our matrix because even if there was a noisy feature, we would just extract a small intensity and place it into our matrix. And by this, the statistical analysis that follows will be more robust. And then we can finally perform statistical analysis. We do an unsupervised segmentation with the spatial shrunken centroids algorithms. And this has two advantages, this algorithm, which was also developed by Kylie, and you can read more on the theoretical parts in this publication. Just to mention shortly, the algorithm is spatially aware. That means that we can work on a spectra-wise level and keep our spatial resolution because the algorithm is able to include the knowledge that spectra next to each other might be quite similar and not independent. And second, the algorithm has also a regularization function that helps to to find the most important features that contribute to the segmentation. And last, we just extract these most important masses for each segment and visualize their spatial distribution. And here I just want to share some more resources with you. So there's plenty of user guides and vignettes on the Cardinal MSI.org webpage. There is many hours of video material in the May Institute playlist on YouTube, and there is a help forum. And with that, I would like to thank the whole Vitek Lab MSI team, especially Kylie for developing and actively maintaining such a wonderful open source software, but also Olga, Dan, and Sai for their contributions to Cardinal and MSI data analysis. And as this is the end of the lecture, I would also like to thank everyone who made this course nice and helped us here. Thank you.
Thank you so much, Melanie. So I think we will just continue a little bit with the hands-on part, right?
Yes.
Or yeah. And after that, we will just finish here so that everyone can have an opportunity to go back to the teaching assistants and ask questions.
Exactly. So I've planned my hands-on part in three, sections. Can you see our studio now?
No, just the slides.
Okay.
Just need to share the entire desktop, I guess.
Yes. Just my RStudio is making trouble. Yeah. Okay. So.
Yeah, we can see it.
That works. Thank you. Maybe we zoom in a bit. So I guess you're now quite familiar with RStudio Cloud and R a bit and how the panel look like here. So I've prepared the analysis of this pig fetus section, and we will now start with the part 1 to load and explore the data. You are welcome to already join me and perform the hands-on analysis with me, but you can also do it later during the TA time. For the exercises, you have to also run parts of the code that I will show now. So you need to install the Cardinal Workflows package, and you can load it with the library function. And here inside this Cardinal Workflows package, because it includes vignettes, there's quite a lot of data available in it. And with the data function, we can extract the data that we want to look at, and this is the pig data. If we run this code, we see that we have 3 different files loaded, but we will only work with the raw data and then generate the pig list and pigs ourselves. So what is often working quite nicely in Cardinal is that we can just run the object and put it into the console. Here we obtain now an error message saying that our object type is MS imaging set and that we should use it as an MS imaging experiment. And this is just because this data set was created in a very earlier version of Cardinal, where there was a different type of our object that stored the MSI raw data. So we can coerce it into the new type of object using the as function. So with this, we coerce the PIC 206 dataset into an MS imaging experiment, and this is the current format how the Cardinal stores the raw data. If we now look at the dataset, we can read that we have a class of MS continuous imaging experiment, and we can now, just to show this, write an IMCML file. And all you need to do is to put your MSI object first. And second, you can specify the name, how you want to call it. So here I have now the PIC 206 IMCML part and the IBD part. And together, those two files are a valid IMCML file. And such files can be read into Cardinal with the read IMCML function. With this function, it's also not necessary to specify the file extension. So I can read it in again and just overwrite the current object. So normally you don't have to worry about this object type, because if you just have an IMCML file and you load it with the read IMCML function, it will automatically be recognized as continuous or process dataset, and you don't need to do any coercion or similar things. So now I would like to explore a bit the raw data and how it is stored and how we can access it. This is just to generally understand a bit which data we are looking at. Many of the dedicated MSI functions in Cardinal do not require to go into this raw data. You can directly specify which masses you want to look at or which functions you want to run. But still, I think it's really helpful to have a look at the raw data. So looking at the masses or features, we can do by using feature data. The short version that gives exactly the same result would be called F data. And when we run this, we obtain a mass data frame. And here we have all the mass bins that are part of the file. If we want to use them directly as a numeric vector, there's a function called MC. And with the MC function, we can check here the class. The class is numeric. So we get a numeric vector that contains all our MC values of the data set. Like for typical vectors, we can perform basic R functions on it. So we can check the length. So we have 10,200 features, and we can also check the range. And the features start at 150 and end at 1000. Then there is this second dimension, the pixels or spectra. As before, instead of feature data, we use now pixel data or P data, which is the abbreviation. And this gives us a position data frame that's basically just a data frame with some specific content already annotated here. So we see a run. One run normally corresponds to one data file. And we have the X and the Y coordinates. The run can also be directly extracted by using the run function. And we see here that we have only one level, meaning that we are currently only working with one data set. And that's what we want to do. And there's also a coord function, which directly gets us the coordinates. So we have X and Y coordinates. And now we come to the third, the third type of raw data that is stored. These are the intensities. We can run image data. Here, the intensity matrices are stored in a list. That's not so helpful. So we can run iData to obtain an intensity matrix, but because this will be quite large, I subsetted the matrix to only obtain the first 15 features, which is rows, and the first 15 columns, which is the first 15 spectra. And then I obtain an intensity matrix. So these are the intensity values. And in the exercises, there's one question about this part of the intensity matrix. And of course, this is all a bit boring to explore, but it can really help to see if there's any issue in the data, or also to know how the data is stored, so that for advanced usages, you can easily find the data and manipulate it. So now it gets more interesting. We also want to explore the visual representation of the image. And I've just picked a random mass, so you can choose different masses, and I've set a plus minus window of 0.2, meaning that everything in this mass window plus minus 0.2 will be plotted as an image. And this image looks now quite boring. Maybe, yeah, it's a bit low intensity in the liver, but otherwise it's really boring. So let's look at the mass 222 in another perspective. We can plot a mass spectrum by using the plot function. So image and plot are just basic R functions, but they are adjusted so that they also work for MSI data in cardinal. And for the plot function, I can specify a certain pixel, and I've just chosen a random pixel number 10. And this is how the mass spectrum looks for this pixel. And here I see that it's at position 70 and 2 in the y dimension. And now I can zoom in into the mass spectrum by using also basic R function xlim. And for this function, the first value here will be the minimum x value, and the second value will be the maximum x value. And if I run this one, I have zoomed in between 220 and 230. And if I now look at the position 222, there's 0 intensity. So it's in this pixel. So probably it's similar in other pixels, and that's why our image looks so boring. We can also plot the average spectrum across all pixels. And this happens if we don't specify any pixel. We automatically run the average mass spectrum.
Melanie, there's a question to you asking,
yes.
what should I change in the code if I want to visualize something with plus minus 5 ppm window?
5 ppm is a bit more tricky, because here we have the absolute value. So you would need to calculate 5 ppm from the value that you want to visualize. So for me, this would be 5 divided by 1 million times 222. So this would be 0.00111. So this would correspond to 5 ppm at mass position 222. But of course, when the value is in ppm, but the plus minus is only in m/c values, this will change for every mass that you want to plot. So I have now the average spectrum here. And again, I see that my 222 is really a bad choice, because there's absolutely no peak. So this is maybe 221.5, which is more interesting. And this is 227.5. So I just draw a line here. I'm not sure if you have seen this before in the course. So with appline, I can draw vertical line at this position, and I can give it a color. I could also change the line type. And if I put an H instead of the V, I could plot an horizontal line. So apparently those two mass positions are more interesting because there's actually a peak. So let's plot the ion images for them. And now we have at least some structure here. It looks, yeah, we can actually see that this mass is not abundant, like negative in the brain and in the liver. And the 227, this is quite ubiquitous distributed, but definitely the intensities are way better because we've actually plotted a peak and not the mass between the peaks. There's also specialized types of ion images, and I think the most popular ones are to plot the TIC. This is the total ion current, and this means that we summarize the pixel dimension and sum up all the intensities for each mass spectrum. This takes a tiny bit longer. And then we obtain for each pixel, for each mass spectrum, the sum of all the intensities that are inside the spectrum. And this is a value that we will later use as a normalization factor. So we will divide each peak intensity by this TIC value, because we assume in areas where the TIC value is higher, probably the whole spectrum is shifted towards more higher values and needs some normalization to become more comparable to the other spectra. And last, for this part, we plot the average spectrum. And this can be done by summarizing the features and calculate the mean. So of course, instead of sum and mean, we could also do different statistical methods and decide how we want to summarize the features and the pixels. And this mean spectrum is actually important for the next part, where we will perform pre-processing on the mean spectrum. And last, we plot the mean spectrum here in this plot. Are there more questions right now?
So far, so good. Please don't hesitate to ask questions if you have any.
Okay, so my original plan was to make a 10 to 15 minute hands-on break. I hope we can still do that. Yeah. You have exercises you can work through with your TA. It's about making some adjustments so figuring out the properties of the PIC 206 data file and to make some adjustments to the image function and explore which here to these functions and explore which parameters are available to customize the plotting of such images. So I think you can now go to your TA. And when should we meet? We have 2 more parts.
So we have overall 25 minutes left. So what would be good?
Let's maybe do just a quick 7 minutes.
Or maybe we can combine 2 parts and just have one longer TA break towards the end.
I have 3 parts.
You have 3 parts.
Probably the longest TA part, I guess.
Okay, let's take 10 minutes. So let's be back at, so it's now 2:50 Eastern. Let's be back at 3 p.m.
Okay.
Okay. And if you would like to stay here and ask Melanie questions,
yes,
don't
please,
hesitate.
if you
right?
have very MSI specific questions, not all TAs are familiar with it. You can stay here and we can discuss it.
I think we can go ahead, right?
Yes. So I hope you had a good time in the breakout room. So I made a smoothed image of the previous plot I had. So here there's many options how you can change the underlying intensities. Just make sure that, remember that whatever you do here to change the image, it will not change your raw data. But it might, of course, not represent anymore the raw intensities if you perform, for example, contrast enhancement and also the smoothing. So we will continue with the mean spectrum. So we have generated a mean mass spectrum for the complete data set. And on this mass spectrum, we perform now peak picking with a signal to noise ratio of 3. So I run this and then I check the object and you will see that there is a process pending peak picking. And this means that by just calling peak picking is not performed, but it is queued to the data set to be performed at a later time point. And this is quite useful because with this, we can chain together several pre-processing steps and run them then all at once. And this prevents us from having to write several objects, which will take some time and also occupy the RAM. So we continue directly and assign the peak alignment to the same object. And here we align all the picked peaks to the mean of the spectrum with the tolerance of 0.5 MC. So this could be also changed. Here we have a unit value, so this could be changed into a ppm unit, and then the tolerance would be in ppm. And this alignment helps to correct small mass differences. And next, we perform a peak filtering. And this peak filtering removes peaks or masses that occur in less than 1% of all spectra. So we just assume if we have a peak that occurs in less than 1% of the spectra, it's probably not relevant. But this, of course, also depends on the type of data you're looking at and on the spatial compartments that are in this data set. And then we also remove all the masses that have a mean intensity of 0. And with this, we actually get way less but more relevant mass features. And now we have queued all these processes to the to our object. And now finally, we can process the queued steps we have added to the object. And this can be done with the process function. And here we get a summary of what happened inside. We can also repeat it and say we want to plot what is happening. And now, because we only work on one average spectrum, we only have one plot here. Otherwise, if we perform peak picking on all the spectra, we would get here a live update of the peak picking of each spectrum. So in blue, we see the signal to the noise line that was calculated. And in red, we see all the peaks that were picked because they had a signal to noise ratio that was larger than 3, how we specified it. And I do not want to go into much detail here, but the process function and all the pre-processing step you can actually run on multiple cores. And this is quite helpful if you have very large data set. You can do a parallel computation and then get hopefully a faster result. We can now also plot the mean spectrum. Of course, we can look at the data set. We have 110 features left. And because we are working on the average, the pixel dimension is only 1. And these steps were just done to obtain the masses that are common to all spectra. So to generate the mass list. And the mass list would then be the masses of this object. So now we can start with the second branch of the pre-processing analysis, which is the normalization. So we use a root mean square normalization. So for the root mean square of each spectrum serves as a factor for scaling all the intensities of the peaks. And again, we need to run the process function in order to actually normalize the file. And now we come to this very interesting step of peak binning. So what we need for peak binning is the profile spectra that were normalized. That was what we have just done. And we need as a reference the mass list, which we obtained from peak picking on the average spectrum. And we need to specify a tolerance. So for each spectrum, at the locations of the MC values from our MC mass list, inside the given tolerance window, the function will look for a peak shape and then integrate the area under the curve of the peak shape of the peak to obtain the intensity value at this MC position. And this takes a tiny bit longer. And again, we need to run process to actually run the peak binning algorithm. And we obtain now a data set, which is 110 features. This will always be the number of your reference. So we had 110 features in our mass list, so we also have 110 features afterwards. After this process, we have again a centroided file, and we have an MS continuous image experiment, meaning that this 110 feature masses are present in all the spectra and stored. Therefore, the mass axis is only stored one in the MS continuous experiments. If we go back to the file after peak picking, we can read that this is an MS processed imaging experiment. And Cardinal by default stores the result from peak picking and following steps in an MS processed imaging experiment, because you would assume that peak picking leads to different mass features in each spectrum. And so it might be beneficial to just store the actual MC values where a peak was picked. But with this step of the peak integration, we are back to the continuous format. And we can plot it again. And that's how it looks like. And with this small pre-processing pipeline, we are now ready. We have peaks of interest and their intensities from the normalized raw spectra, and can then continue with the segmentation part. So I will run this first because it takes a while. So what I do here is I set a seed, and this is necessary because the spatial shrunken centroids algorithm performs a random cluster initialization, and this is based on a random number generation. And if I would not set the seed here, then it might happen that the next time I run the code, I get a different result. So the function spatial shrunken centroids takes as an input our pre-processed R object. It then enables two types of methods for smoothing the neighboring pixels. And I have chosen the adaptive smoothing because the adaptive smoothing smooths the neighboring pixels, but not so much when there is like an edge, meaning that there's a different histology in the neighboring pixel. It will recognize it and not perform a smoothing on this one. And the R is the smoothing radius. So we perform a bit of smoothing for the 2 neighboring pixels of each pixel. And for K, we set the initial number of clusters, but the segmentation might end up with less clusters than that if it fits. So this is really just setting a maximum value. And S is a sparsity threshold by which we shrink the T statistic. So the higher we set S, the less features will become part of the clustering algorithm. And the more we obtain the features that really make a difference between the compartments. But which number is ideal for a data set is really data set dependent. And so it's always good to choose several values for S and see which one looks good for the data set. We obtain then an object, a results object here, and we can run a summary of it. And this gives us now a summary for each of the segmentation we have performed. So R and K were always the same, and the shrinkage values were different. And here we can recognize that for higher shrinkage values, we obtain less classes based on less features. And then we can look at how these clusters look like in the image representation. I just have chosen 3 of the S values here, but we could also plot all of them. So we see that for the S equals 0, we have quite distinct clusters here. It looks a bit noisy. The liver and the heart are already nice. But here with S equals 20, we have less clusters, and we have here a region for the brain, for the heart, for the liver, and here for the back. So we have a closer look at the clustering with S equals 20. And from this color code, we already see that we have 6 clusters. That's also what we have obtained here in the summary. And now we are interested in also seeing how the mass spectra look like of the different compartments. And this we can do again with the plot function. And the plot function gives us now the centers, and this means the average mean values for each of the clusters. And if we look here, so the heart was the pink cluster, and for the pink cluster we see here two peaks that seem to have a higher mean intensities than the other clusters. Then we can also plot the T statistics by saying we want to plot the statistical values. Here we use just a different line width. And here again, we see 2 pink mass features for the heart and many distinct statistical values for other masses. And the T statistics, the higher the T statistics is, the more important the mass feature for this cluster. And due to the shrinkage we perform here with S equals 20, we actually shrink T statistics towards 0. That means if a mass feature had only a small intensity or a small contribution towards the cluster, we would shrink it to 0 and remove it from the clustering because it made only no difference or just a very tiny difference. And this gives us in the end really the most important features which are plotted here that enable us to distinguish the different molecular composition. And now at the very end, we would like to look closer at the heart section here. So from the color, I can say that the heart is the class number 6. So I can extract the top mass features from the results data set for s equals 20 and class number 6. And looking at this, here's are the masses. R and K were always the same, and S I now specified at 20, so it's also the same. And of course, then we also have the same classes because we only look at one segmentation approach. And for each mass, we now have the average intensity here in the center column, and we have the T statistic in the statistics column. And actually, for the heart, we only have 2 masses that have a positive T statistic. So only the 2 masses that we have seen in the spectra before contributed towards the segmentation of the heart. And of course, I would now like to see how the mass looks like. So I plot an image on the actual really raw data for the mass 187.3. I apply a small window to it. And now I have the distribution of this mass that obviously is more abundant in the heart region. And if we have a little bit time for exercises, one of your exercises would be to repeat the pre-processing and the statistical analysis and then find out which cluster corresponds to the liver and to the brain and extract the top features and plot the visualization, so the image of the distribution of your top features in R, exactly how I have done it here for the heart. You could do it for the liver and for the brain to find the most important mass features and visualize them here. Are there questions?
Well, so far, no questions, but I think we are running out of time now. So what I would like to do is to thank Melanie and thank all the instructors in the last 2 weeks, and also thank the participants for sticking with us, because it is really tiring to do this type of work on Zoom. And I would very much like to thank the teaching assistants. So we were kind of trying this new way of providing hands-on assistance and personalized assistance. And I hope that it was useful to you today or maybe in the previous days as well. So we're very grateful. Most of the teaching assistants are masters in data science students at Northeastern, and we have some TAs who are also from other institutions. So we are very grateful for their contribution. And maybe I'd also like to say that I hope that you have appreciated the different level at which we can work and interact with R. It can be really base R, doing some really basic plots, or we can use packages which already exist, such as ggplot. So they're written within the R community and they provide more advanced capabilities for data visualization or data manipulation. But we can also write our own packages, like we have seen towards the end of this 2 weeks, which are specific for our community, specific for the types of problems that we're dealing with. And so there's a lot of opportunity for us to improve on that. And so the value of R is in part in that we can build on the existing packages, but we can also open them up, we can look inside, and we can extend them. And we can develop new both statistical methods and implementation. And so I hope that you will continue looking into that, either as a user, or maybe some of you would be interested in contributing some functions or some code for some of the problems which are specific to your work. So thank you very much. We will send you a questionnaire on behalf of ASMS about your experience in the last 2 weeks. Please kindly respond if you don't mind. And if you're interested in learning more, please watch out the space, especially on our website, and join us at the May Institute and maybe in other short courses. So I know we will be doing another short course at ASMS associated with the main conference in June as well. So thank you very much. So Melanie and I will stay here for a few minutes in case you have questions. And if not, we hope to see you around. Thank you again for joining us. And thank you for the nice feedback. So I will stop recording already.